Um, yeah, so when I was invited to give this talk, I was a bit confused because I don't really work on viruses outside of phage. Um, and I decided I'm not going to tell you about those today. So instead, I want to tell you about a project we've been working on recently and something we've been thinking about, just because I think it's interesting and it's changed how I see the whole game of microbial evolution. And so hopefully you'll find this interesting. I always like starting by arguing to microbiologists, and I think probably to you this is unnecessary, why you should care about evolution. And I think, you know, really it's core to microbial life. The great example that I like from, I think it was Roberto Coulter, was if you start with a single E. coli cell growing at exponential phase, like many of you who have any lab experience have, you'll know overnight or in about 13 hours, you get a liter full of bacteria. And if you keep going for about 27 more hours after that, it actually, assuming you ignore the laws of physics, a single E. coli is now the size of the entire Earth. And 16 hours later, it hits the Schwarzschild density. But the this is obviously impossible, right? Because of physics, chemistry, and any number of other things. But the point is just that microbes that are capable of rapid generation time, whether they be bacterial or viral, can really fill any ecological niche really, really fast. And this also means, if you have a population and there's just a few little weirdos with mutations, if the environment changes or if they end up migrating to a new environment, they can fill it very, very quickly. And of course, we all saw that a few years back, probably part of why we're here, right? This famous tweet on January 5th, 2020, with a total of 44 cases of a brand new pneumonia. We all know precisely how that went and is still going. And I came to this from a different angle through antibiotic resistance. So antibiotic resistance, you usually think of it as like the canonical Darwinian evolution. You have a sensitive population with a rare resistance cell. Antibiotics kill everything but that cell. It spreads. Great. But to really try to predict it, to try to make a difference in it, you got to look closer and ask how it happens. And there are two primary ways it happens. The first is spontaneously, that there's some error in replication that allows a cell to be slightly more resistant to antibiotics. In my postdoc, I worked on this, so I built this giant petri dish to see how this happens. And you can see over the course of about 11 days, you can evolve super high levels of resistance. So this is bacteria growing in an increasing concentration of trimethoprim as you go towards the center, and you can see them, literally the whites bacteria spreading out, hitting a new boundary, and then that selects for the members of the population that have those rare mutations. And this was really exciting, but the more that I looked at this, the more I feel like there are a couple things that are misleading about this. The first one is you look at this and you think, wow, resistance can just evolve immediately. But you actually go into the clinic, and of course, drugs still work all the time. This isn't what's happening in the clinic. And the second is that this is not really how most resistance evolves. The main way that clinically relevant resistance happens is by horizontal gene sharing. So this is something you could never see pop up like that in the lab because it's these very specialized resistance genes that get shared by other microbes, oftentimes on plasmids. So any of you who have done any amount of lab work have probably used the plasmid for, for cloning, for introducing genes, for doing things. People basically come by plasmids two ways, either that or in the clinic worrying about antibiotic resistance. And I hope what you get from this is that there's actually a lot more biology going on, even with these things we think of as normal tools, because when you look at what they are, So they're often multi-copy, sometimes a single copy. They're self-replicating. They carry all sorts of interesting things, but the fact that they are self-replicating and transmit between different, can transmit between different bacteria means they themselves are evolving, right? Plasmids that can spread better, that can replicate better, also do better. So we started looking at this from a couple of angles. First, you know, we did, I guess, the medical school equivalent of what naturalists do, which is we just looked in the hospitals. So my postdoc Celia found this great data from, of all places, the US Army. So they sampled every E. coli from Walter Reed Hospital for an entire year. And I think what I want to draw your attention to is over 70% of the E. coli that they found carried a plasmid. Over 30% carried this one particular plasmid that's yellow here. Interestingly, that plasmid does not carry antibiotic resistance, so it's successful for some other weird reason. We also find in other collaborators plasmid outbreaks in hospitals. So these are, different patients in the same ward with different pathogens that share an antibiotic resistance plasmid that's precisely identical to the nucleotide. Looking historically back from samples as far back as 1918, it seems to be increasingly mobile. So there's plasmids are emerging and changing. And so we asked this question, and I want to just, I guess I've got a couple more minutes, tell you some of the experiments we're doing to try to understand this question of how do new plasmids emerge. So what happens is, particularly with multi-copy plasmids, so first you get a new one that pops up, and the first thing it has to do is it has to outcompete its neighbors within the cell to take over that cell. And then that cell needs to take over the whole population. And so the second is what we usually worry about when we think about the cargo. The first is what we're worrying about. This has been historically really hard to measure because to understand what makes a plasmid more fit than another, you have to do this crazy thing where you make a huge population of cells with an exactly balanced number of plasmids of two different types. And then all at once you have to say, okay, stop balancing, let it compete. I thought this wasn't doable until postdoc in my lab proposed this idea. Fernando had this idea that I thought this, and I'll admit, I thought this would never work. I told him this. I was like, okay, you've got 6 months to try something, but then we're going to do something else. Because he had this idea of let's take two random plasmids, fuse them with a recombinase site, and then find some way to switch on a recombinase on command that'll pop them into two. This took an incredible amount of work. In the end, we had to go find a yeast from Patagonia that has not seen metabolic temperatures for 50,000 years, use its flip recombinase that turns out not to work at 37 degrees, but works beautifully at 30. And so now we can basically take these dimeric plasmids, grow them up inside cells, and then take things out of the 37, put them in the 30, and then they start competing. So what do you expect to see? Well, naively, if you think about human chromosomes, you might think, all right, we'll just see the same thing in each one, but that's not what goes on because there's not a segregation system for high copy plasmids. What you really see is that this one segregates sort of randomly, and then those randomly repopulate, which then segregate randomly and randomly repopulate. And if any of you have taken population genetics, this should look really, really familiar. This looks like, the Wright-Fisher model or something like this. And I want to show you what this actually looks like inside individual cells. So this is actually a photograph of a petri dish. It's a black petri dish. The cells, the two halves of the dimer are identical, except one has a red fluorophore, the other has a blue fluorophore that are almost precisely identical. And in this picture, we've just taken them out after 12 hours of growth. We've just taken them from the 37 and we've put them in the 30 under a camera. And so I'm going to show you a time lapse of what happens. And so what you can see really is that at the end, these brightly colored things are cells that have the same that are sort of fixed, where one or the other is taken over, and that in this interim, there's competition between the two. Of course, for those of you who might be worrying, oh, is that yellow actually red and blue cell? Like, are they actually yellow cells, or are they just red and blue on top of each other? We did the control, and in fact, yeah, it really is. And what this means is that in this yellow area, there's time for competition. I'm going to go very quickly over this. So when we do this splitting, it actually at a very low transient rate re-dimerizes. And what's great about that, it means we can sequence across the junctions to sample how diverse individual cells are, because it's sort of randomly joining two plasmids inside every given cell. And then we can sequence across to see what fraction are red-red, what fraction are red-blue. So at the beginning, you expect, okay, they should all be different, right? Because they're the dimers. Then we split the dimers and you expect, okay, it should be 50-50. And then there should be some sort of exponential die-off. And what was beautiful about this was that actually that's exactly what it looked like. Yeah, Max, I can see you smiling. This is one of these rare moments where you draw the plot and like, oh my God, it looks like that. But what's even better is it's actually a real exponential. So segregation is memoryless, and I don't have slides on this, but one of the things that was really interesting is when you try to model it, you actually find that all the models give you much steeper die-offs than you expect by chance. And it turns out what's happening is that the plasmids have this system that slows replication after one of them replicates. So sort of after a plasmid duplicates, it pauses for a bit before it can duplicate again. And that slows drift down. It basically means at that second step here, this refreshment is more quantitatively faithful. And we don't know why that's happening. Our best guess is it's the cell basically domesticating plasmids. Because when we take it out, plasmids do better. But also, when we take it out ourselves, now we can really look at what makes plasmids replicate better. And the last thing I'm going to tell you about is we found one of the things that makes plasmids worse is expressing a gene. So here, we compete strong red versus weak blue, strong blue versus weak red, or you know what I mean, both combinations. And we find is whichever one is more transcriptionally active loses. Now, you may think, oh, well, of course, it's costly to produce a protein. So we've actually been looking at single cell dynamics, so we can get rid of that by using a device called the mother machine. This is a microfluidic device that isolates things inside trenches. What it lets us do is it basically, as cells divide, whichever one's on top always stays in. And that means if one or the other offspring divides faster, it's irrelevant because you're just picking a random one. But plasmid dynamics still happen within it. And this gives us some really pretty movies, you know, of all of these things evolving. Oh, there we go. Right, you can see all of these things evolving inside, or really competing, not evolving yet. And the point is, it's irrelevant who wins. Because it's microfluidic, we can do 1 million replicates at the same time, which is, you know, like 10,000 times more than you actually need to do the experiment, but who doesn't like overkill? And when we do this, what we find is for the same promoter, you'll see the red does a little better. This is actually because of an extra hemimethylation site we didn't realize. But when we put a weak and a strong promoter, again, we see really, really strongly that the strong promoter plasmid just doesn't replicate as well. These are the error bars on this. And so I think there's this really interesting thing happening where, I mean, we don't have solid conclusions of this yet because this is still very much in progress. But my best guess, if I had to say what's going on, is that because plasmids have to both replicate and express genes, that doing both of them interferes with each other. Like the uncoiling to express a gene induces supercoiling that prevents binding of the initiation primers or something. Some sort of interference is happening. And this means plasmids on their own are pretty bad at sort of carrying genes. Or a gene gets into a cell that's beneficial, the cell doesn't really have time to see whether it helps before that just dies off. And so my best guess is that these methylation systems that pause replication and suppress competition are really there to help the cell sort of sample genes, to allow new plasmids that take on a new gene to have a better chance of emerging in the population. All right, I've run way over my time. I'm sorry about that. This is the lab, funding sources, and I will be happy to take questions in whatever time I have remaining. I hope that, at the least, the next time you use a plasmid, you look at it a little differently. That it's really its own evolving entity, despite being just a couple thousand bases of DNA.